Now see what's written over here. Since we have done the primary, secondary, and tertiary structure, we haven't done the quaternary structure over here. But remember, you are asking me a question on sickle cell anemia.
Oh yes, Miss Sickle.
Acha, so what actually happens in the sickle cell anemia is that sickle cell anemia, you've been studying from O levels that it is a disease which occurs due to a mutation in the gene that controls the synthesis of hemoglobin, isn't it? It's a mutation. अच्छा, अब वो mutation gene के अंदर होती है, and mutation in the gene का मतलब कि there is some defect in the DNA. So if there is a defect in the DNA, there will be a defective protein made because it's actually our code in the DNA that is controlling protein synthesis. ये बात समझ आ रही है?
Yes.
या नहीं आ रही? अगर नहीं आ रही तो मुझे साफ बता दो कि नहीं आ रही, because you see many students don't know it because maybe उनको O level में नहीं बताया हुआ होता है. So I'll just give you a very brief idea कि all of us know that we have trillions of cells, and in trillions of cells we have a nucleus, and in that nucleus we have a DNA. ठीक है? DNA determines what we are, and DNA is a code or a set of instructions. Just like a computer code, it is a set of instructions. क्या मतलब कि it's a set of instructions? It's a code which actually, it's a code of certain letters. I'm not going to go into the details of that code right now, but that code determines, और एक नहीं, हमारे पास पता नहीं कितने millions of codes हैं. Every code determines what kind of a protein will be synthesized. What kind of a protein will be made. For example, there is a code, अभी मैं आपको समझाने के लिए बता रही हूँ. Let's say there's a code A, T, C, G, C, C, T, 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 A. This is a code, and I can't understand this language, but basically this code inside your body is translated into a language, right? And that language is that of what? उस language को हम कहते हैं primary structure. ठीक है? Primary structure, you know, right? What is a primary structure?
It's a chain of polypeptides, miss. Amino acids.
बस सिर्फ chain of polypeptides या कुछ और भी? Is this a primary structure?
Chain of linked amino acids, miss.
हाँ, a chain of amino acids arranged in a particular sequence, और हमें नजर आनी चाहिए वो sequence, ठीक है? So the code in our DNA will determine what kind of protein is made in your body. In your body, amongst the long list of proteins जो हमारी body में है, one of the protein is hemoglobin. ठीक है? तो इसका मतलब ये हुआ कि there must be a code inside your DNA which determines कि अच्छा ये वाला molecule synthesize होगा क्योंकि इसकी sequence of amino acids ये है, right? Now, if there is a problem in the DNA code, in other words, if there is a mutation or a change in the DNA, do you think that the primary structure should also change?
Miss, if the DNA code is changing, then yeah, it should also change the primary structure.
It should change
because then the sequence of amino acids will change also.
Exactly. Okay? And the same thing happens in sickle cell anemia. There is a change in the DNA code, which is nothing but a mutation. If the DNA code changes, then of course the protein that was responsible to be made from that code. For example, ये code ये था जो मैंने अभी लिखा है. मैं इस code को change कर देती हूँ. Okay, let me change this code. I'm erasing a letter and I'm putting the letter A. This is the mutation. ये मैंने mutation बना ली. तो अब, of course, when I change this, I will not get the same sequence of amino acids which I was getting before. Okay? So this means that any change in the DNA code will change the primary structure of the of the polypeptide, and hence it will change the hemoglobin also. इसी तरह की एक mutation होती है sickle cell anemia, in which the DNA code has been changed in such a manner कि एक chain है amino acids की, ठीक है, और वो chain जो है वो बहुत सारे amino acids long है. I think 141 to 146 amino acids है. उनमें से एक amino acid होता है glutamic acid. Okay, It is replaced by a non-polar amino acid which is valine. ठीक है? Glutamic acid है आपका polar and it's replaced by a non-polar amino acid. You tell me, what do you mean by polar amino acid? Will a polar amino acid be soluble in water?
No, Miss, it won't be soluble in water.
Polar amino acid.
Polar, Miss, polar, it will be soluble.
And non-polar?
Miss, that won't be soluble.
That will not be soluble. ठीक है? तो अब देखो क्या हो गया. Just by changing one amino acid, and specifically what amino acid, ये आपको याद रखना पड़ेगा. Glutamic acid, which is polar, is changed or replaced by valine, which is non-polar. So since अब पूरी जो है ना स्ट्रक्चर ही चेंज हो गया हीमोग्लोबिन का. Glutamic acid ने क्या किया था? Because it was polar, so hemoglobin had to be soluble. ऐसे soluble, इस तरह से soluble. देखो, hemoglobin is a protein and it has a primary structure, tertiary structure, and a secondary structure also. This is the tertiary structure that we did last time. हमने क्या कहा था कि tertiary structure is extensive folding and coiling because of disulfide groups, because of hydrophobic interactions, ionic bonds and hydrogen bonds. ठीक है? इसको इमेजिन करो कि ये वो है, हीमोग्लोबिन है. ठीक है? हीमोग्लोबिन fold हो गई into a tertiary structure, into a ball shaped structure, and I'm talking about the normal वाला hemoglobin. ठीक है? I'm not talking about sickle. तो normal वाले hemoglobin ने क्या किया कि all the non-polar amino acids, they group themselves in the center, well away from the aqueous environment of the body, and all the polar amino acids, they were facing the exterior or the aqueous amino acid, making the hemoglobin soluble in water. ठीक है? अच्छा, अब हम आते हैं सिकल सेल में. सिकल सेल में क्या हुआ कि ये जो थे आपके सारे पोलर, ठीक है, ये बन गए अब नॉन पोलर. नाउ व्हेन दे बिकम नॉन पोलर, व्हाट विल हैपन? चेंज हो जाएगा शेप हीमोग्लोबिन का. ठीक है? क्योंकि ये इन पोलर अमाइनो एसिड्स की वजह से ही इस मॉलिक्यूल ने ऐसे करके कर्ल किया था. इस तरह से कर्ल किया था कि सारे नॉन पोलर अमाइनो एसिड्स अंदर आ गए थे और सारे पोलर जो है वो बाहर चले गए थे. सो since glutamic acid, why is my voice echoing? Okay, since glutamic acid is replaced by valine, so what happens is it makes your hemoglobin less soluble as compared to when it was the normal hemoglobin. ठीक है? आपका hemoglobin less soluble हो जाएगा and the shape of the red blood cell will also change. Normal hemoglobin, the shape of the red blood cell is a biconcave disc, or sickle cell hemoglobin may, because of the insolubility of this hemoglobin molecule, the shape of the red blood cell also becomes sickle. Are you understanding this phenomenon?
Yes, Miss.
ठीक है? अब आप इसको रिपीट करो अपने वर्ड्स में. What did I say? Repeat this.
Miss, basically, there's a mutation which leads to a change in the base sequence of DNA. So the arrangement of amino acids changes. So in the case of sickle cell anemia, because one amino acid is changed, glutamic acid is replaced by valine. So the composition changes in a way that um, from a polar thing, it changes into a non-polar substance. Miss. So the coil gets disturbed because earlier the non-polar regions were facing inside. They were facing inside,
Yes,
and
yes.
the polar regions were facing outside. So now, since there are no polar regions, everything will face inside.
This is going to happen. Read this. Right?
Yes, miss.
अच्छा.
So, miss, if
Yes, beta.
since there are no polar substances, the coiling is affected, so it will now become a straight chain, right?
It can become a straight chain or it can become straight chain की वजह जब straight chain बनेगा तो shape of the red blood cell also changes. So basically it goes like this. To summarize, I can say a change in the DNA changes this base sequence, and a change in the base sequence changes the primary structure of the protein, and a change in the primary structure changes the tertiary structure of the protein. Is that making sense?
Yes.
Okay. So this is, that's why you can now understand how important it is to know the correct concept of a primary, secondary and tertiary structure. अब इसको आप देखो गौर से. You have a DNA code, right? और इस DNA code ये कैसा था? ये बिल्कुल इसको कोई रटा लगाने की जरूरत नहीं है, but very simply इसी ये था CAC और GTG. तो सिर्फ इसकी इस changing की वजह से what happens is that instead of glutamic acid we have valine. Now glutamic acid was polar, valine is non-polar. When you have glutamic acid, your hemoglobin is more soluble and your red blood cells are biconcave discs. When you have valine, your hemoglobin is less soluble and the red blood cells shape is sickle. So hence the mutation. Very commonly asked question. What could be the, acha nahi, you can't do this because you haven't done quaternary structure. Let's start doing the quaternary structure. Read these bullet points and then we'll move forward.
So miss, this one does not have the disulfide bond?
This has everything. A quaternary structure का मतलब ये है, usually it is tertiary proteins. Usually all the proteins which have a tertiary structure, सारे bonds उनके पास पहले से होते हैं, उनका एक quaternary structure भी. इसमें से फर्क ये है कि it has more than one polypeptide chain. जैसे कि मैं आपको इस तरह बताती हूँ, देखो ये जो आपको डायग्राम बताया था ना, how many polypeptide chains can you see in this diagram? Polypeptide chains, backbone की बात कर रही हूँ. How many backbone polypeptide chains are visible in this diagram?
Is this one chain?
Very good one. अच्छा, अब एक और डायग्राम आपको दिखाती हूँ. How many polypeptide chains?
Miss, this is also one.
This is also one. Very good. Now another one. How many polypeptide chains? Chain 1, chain 1, chain 2, chain 2. कितने हैं इसमें?
So miss, 4 chains?
Four chain. So this is your hemoglobin molecule. ये है आपका hemoglobin molecule. And you're not supposed to remember all this. बस इतना पढ़ना है आपको, याद रखना है कि this is your red blood cell. ठीक है? If we cut open a red blood cell, we'll see millions of hemoglobin molecule. और अगर हम उनमें से एक hemoglobin molecule को निकालें, so every hemoglobin molecule is made up of a combination of heme and globin. ये जो globin वाला part है ना hemoglobin का, that's actually the protein component, protein. And globin wala part is made up of 4 polypeptide chains. 4. What are those? 2 alpha chains and 2 beta chains. Alpha chain, beta chain, beta chain. Now I'll ask you a question. Arya, tell me, do you think that within these chains you can see a primary structure?
Miss, in primary structure, we're basically seeing the sequence of amino acids, but we can't see anything here.
So there's no primary structure. Secondary structure नजर आ रहा है?
Miss, there's a helical structure there.
There is. And can you see tertiary structure? Has it been extensively folded into a ball-like shape?
Yes, miss.
And can you see a quaternary structure also?
Okay.
Quaternary की क्या definition थी? Any protein that has at least more than one, तो इसके पास तो चार हैं.
Yeah, miss. So we can see the quaternary. Yes.
तो इस diagram में you will say I can see the secondary, tertiary, and the quaternary structure. And for each thing you will give a reason. And you will say I cannot see the primary structure because I can't see the amino acids arranged in a particular sequence. उसके लिए मुझे amino acids के नाम नजर आने जरूरी हैं. Is that clear?
Yes, miss.
Now let me show you some other quaternary protein. ये तो नहीं है. Again, can you see the quaternary structure?
Yes, miss. I can see.
How many polypeptide chains can you see?
Miss, 4 chains.
4 chains. अच्छा, अब इसमें गौर से देखो, ये सारे जो chains है ना, those are the globin part of the hemoglobin. और जो heme part है ना, that is red in color. ये आपको नजर आ रहा है heme part? That is red in color. And why has it been stained red? Because it's actually the heme part which contains iron atom, Fe+2. So we say that a hemoglobin molecule is made up of the heme part and the globin part. The globin part is made up of 2 alpha chains and 2 beta chains. Okay? The heme part is the non-protein component of the hemoglobin molecule. I repeat once again, the heme part is the non-protein component of the hemoglobin molecule, and such a kind of a non-protein component, which itself is non-protein, but it's an essential or a vital part of a molecule, is known as a prosthetic group. इसको प्रोस्थेटिक ग्रुप इसलिए कहते हैं कि इसका होना बहुत जरूरी है, लेकिन ये खुद प्रोटीन नहीं होता. What's the importance of this prosthetic group? The importance is that it carries 4 iron atoms. We have got 4 heme groups in one hemoglobin molecule. Each heme group has 1 iron atom, and each iron atom can bind to 1 oxygen molecule. So in total, 1 hemoglobin molecule can bind to 4 oxygen molecules. Is that clear?
Yes, miss.
Okay. So I'll just give you some details and you can note it down. For example, if I have, I would say like this, 1 hemoglobin molecule sorry, I've made a mistake over here. 4 heme groups.